right? We can see basically two changes, that is increase and decrease. We can take production of interferon as another important thing. So then I start production of interferon start, because interferon produces a protective mechanism against viral infection, and the massive increase is due to large cells infected with the virus, and all the cells start to produce now interferon. Interferon levels goes up, and then start to decrease, because when virus are cleared by the body by the immune system phagocytosis, then virus cannot infect more cells, and then it decrease. Now we can also think, should we write what is the role of interferon? If you want to write it here, it is only this place you can write, because interferon can interfere with the virus cell cycle. It produced due to a warning signs to the neighboring cells, and virus replication could be reduced or attachment reduced by the neighboring cells. But 3 marks, maybe you spend 3 minutes. So you can decide what extent you have to write, as time management is important. No interferon produced while virus attached. Increase in interferon production because infected host cell produce interferon. More cells become infected. Interferon produce falls as viral infection prevented. That the interferon produced falls as virus particles are destroyed by immune system. So it doesn't expect all the roles of interferon. They ask only changes. We see one change: start production, increase and decrease. So they ask here, marking scheme wants to know why it is important, and not expecting a role of interferon. The immune system to viral infection also involve T cells. Distinguish between the role of T helper cells and T killer cells in the immune response to viral infections. I think still you need this, because frequently asking question. And the role of T killer cell, they are very important in virus, cancer, and sometimes transplantation. Because transplantation organ consists of cells. These cells are destroyed by T killer cells. Cancer cells are destroyed by T killer cells. Viral infected cells are destroyed by T killer cells. That means it destroyed not the virus, it destroyed the infected cells, virus infected cells, cancer cell and transplanted cells. And further, we should know how they do it by secreting enzymes, because T killer cell identify these cells. They express this antigen with MHC. This is recognized by T killer cell, and then T killer cell secrete enzymes. Usually, new marking schemes, the name of enzymes are not much important. Example, perforin is one of the enzyme. And perforin make holes on the membrane and cell destroyed. Then the virus come out. Phagocytic cells destroy the virus. That's the role of T killer cells. Okay, now T helper cells, after identify the antigen by antigen presenting cell, MHC, and the antigen, this is identified T helper cells, and T helper cells undergoing mitosis, and it become effector cells. Then they can activate T killer cells and B cells. So you write here T killer helper cells are important for the activation of T killer cells, and after antigen presentation, they increase number by mitosis and become effective and activate T killer cells. Activated T killer cells can effectively destroy virus infected cells for phagocytosis. T helper cells T killer cells and B cell, where T killer cells host infected cells, virus infected cell destroy. T helper cell produce cytokine, whereas T killer cell produce perforins, chemical enzymes. T helper cells involved in the humoral, whereas T killer cell involved in cell-mediated response. Cell-mediated response is one which T helper cell is doing, and mostly humoral immunities, B cell is responsible, and here they accept here humoral is also okay for T killer cells. Phagocytosis, it's a non-specific response and it's an immune response. Destruction of pathogen. So which one you think? So destruction of pathogen start very early by the phagocytic cells. And this is non-specific. After that, it goes to the specific immune system. Specific immune system produce antibodies. So these antibodies important further increase phagocytosis. So that's a specific immune response. Non-specific response initially, after that, specific response. So we can still consider destruction happen is started with non-specifically, even interferon important here, they're non-specific, and phagocytosis started early, non-specific, but later specific immune response further enhance it. So destruction of pathogen, we can take this antigen presentation. So antigen presentation, it happened in this specific response, antigen present here. So we can consider it as an immune response. So I quickly highlight again these things as you know, needed MHC and antigen, and this cell become a antigen presenting cell. That means phagocytic cells, if they show antigen and the MHC, it become antigen presenting cells. Antigen presenting cells enter the lymphatic systems, move to the nearest lymph node, present antigen to T helper cells. Then this is antigen presentation. After antigen presentation, T helper cells undergo mitosis or clonal expansion, and they increase the number of T helper cells. Then T helper cells, they can activate T killer cells by giving a cytokine signal, and they can also activate B cells by giving cytokine signal. T killer cell can destroy virus infected cells. T killer cell can destroy the cancer cells. T killer cell can destroy transplanted cells. B cells act as antigen presenting cell. B cell phagocyte pathogen, display antigen. That means B cell is antigen presenting cells. Present antigen to T helper cell, cytokine signal. B cells activate and undergoing mitosis and produce plasma cells. Plasma cell produce antibody. Then antibody has several functions: agglutination, making all the pathogen into one place by making this network, and opsonization. That means they tag the pathogens and bring to the phagocytic cells, and toxin neutralization. So these are the three main functions of antibodies. And this is the structure of antibodies. They have two heavy chains and two light chains. Red color one is light chains and disulfide bridges, and two identical antigen binding sites, this and this. Okay, do you have any informations, anything to ask, Virus?
No, sir.
Then we can go to the forensics. The forensic main aim is to find the time of death. So there are many ways this can be asked. Calculation, a question of time of death, and different method to use time of death, and different techniques used to time of death, like many things. So if you are get any kind of a completely new thing, don't be panic, because at the end it's not that difficult. For example, uh, this is one question asked. One method is to use a Henske nomogram. Now, when you see such thing, don't be panic. Just uh, check the question. The Henske nomogram relate the time of death to the ambient temperature, core temperature, and the mass of the body. Because temperature drop is related to the body mass, body clothing, ambient temperature, and then um, where it find the place where it find, like inside or outside, like many factors are important here. At least they can check body mass, core temperature, and the ambient temperature. Explain why the ambient temperature and the core temperature of the body are used to determine the time of death of a person. Because after death of a person, core body temperature decrease. Core body temperature, this decreased core body temperature depend on the ambient temperature. So if the person, let's say like this, so core temperature is 37. This 37 is really important. Let's say outside temperature is 0. Outside temperature is 37. Now you see, in this case, heat loss is faster. Body rapidly cool down, and this case, body cool down is slower. Then we need a method of the rate of heat lost for each temperatures. And then heat loss is very important factor. It is based on the body size, volume, and surface area to volume ratio. So people develop certain method now combining all these things. Okay, here man is found murdered on, okay, not that one. The chart shows Hensege nomogram. The steps below need to be followed. 1, 2, 3 is given to you. Draw a straight line between this and that, and draw a straight line again and read the temperature. Now it's like this. This is a Hensege nomogram. First thing you are asked to do, draw a straight line between the core temperature of the body and the ambient temperature. Now here is the core temperature. You can see core temperature here. Where is this pen? Okay, here, core temperature. And this side, ambient temperature. So ambient temperature is given 0 to 20. So draw a straight line between the core temperature of the body and the ambient temperature. Okay, now here the person, it is given in the question, the temperature is 25. Core temperature is 25, ambient temperature is 15. So we can draw a line from core temperature to ambient temperature. A second one, draw a straight line that extends from the center of the circle through the diagonal line at the point where it crosses line 1. Okay, this is a diagonal line and this is a line 1. It crosses here. This is a crossing point. Then we have to draw a line from the circle, center of the circle, to this crossing place, like this. And then read the time of death from the nomogram at the point line 2. Line 2 is this one. Crosses the appropriate semicircular for the mass of the body. The person body mass is 100 kilogram. So then if it is crosses 100 kilogram at this point, it is 22. It gives 22 hours. The time is 22 hours is the time given. So that happened 22 hours before. Here's the question. A body was found. The mass of the body was 100 kilogram and core temperature of the body 25. The ambient temperature is 50. So we did the same, 25 to 15. And next crossing point, and we find 100 kilogram is the mass of the body. So the idea is you can ask time of death in many different ways, pure mathematical calculation, and this kind of different methods. So don't panic, just read carefully and then try to find accurately. Okay, if the body is neck body lying stretched out and in a still air, suggest how change in each of these three factors could affect the estimated time of death. Give a reason for your answer. Now, neck body increase heat lost, so then bodies rapidly cool down. Lying stretched out, so lying stretched out is more space to heat loss, and again quickly cool down the body. The still air is not moving, then heat trapped with the still air. This helps to reduce heat, cooling down of the body. So factors are very important when we going to write answers. All factors you consider carefully. Sometimes we don't learn all the factors, but you need to know whether these factors are contribute to heat loss or heat reduction of heat loss. Okay, that's it from forensics. Yes.
So when the body is in still air, that means it doesn't reduce the body temperature, right?
Yes, I think this is typical question asked. I mean, this question is based on plant transpiration, because transpiration is loss of water from the plant. And if there's a wind, transpiration is high, because if the body is like this, And if wind blows, wind can take this warmer air out of this body. If the still air mean the air is still warmer here, so then the heat loss can reduced. So wind speed also affect the heat loss. Still air keep the warmer air around the body and it's not moving, so heat loss is less. So any factor carefully consider how it affect for the cooling of the body. Do you have any more questions about forensic?
No, sir.
Maybe there is a question, maybe there is no question in forensics. This happens sometimes. The forensic question is not always coming, but climate change, we get more than one question. I didn't bother about the photosynthesis because photosynthesis, I hope you can manage. It's not that difficult part because it has a little bit less application. I try to get some other questions. Then next one is antibiotics. The effect of three different antibiotics, PQR. So antibiotics are chemical which can kill or which can reduce the growth of microorganisms, bacteria. This PQR investigated three antibiotics. Small disc of filter paper were each soaked in different antibiotic and dried. These discs were used in this investigation. The flow chart below shows the procedure used in this investigation. Suspension of bacteria. Bacteria spread on the surface of the agar in two petri dishes. So we can spread the bacteria equally, two petri dishes. Petri dish 1 incubated at 30 degree for 24 hours. Then we can place this petri dish 30 Celsius degree 24 hours. And for this one, antibiotic disc placed on the surface of agar in petri dish 2. Then this step, after incubation, they added the disc and again incubated 30 degree for 48 hours. And this one straightforward 30 degree for 72 hours. So at the end time is same, here 24 and 48 and here 72 and here is 72. The only difference is one dish incubated with bacteria, then disc added. Appearance of petri dish 1 after 72 hours, appearance of petri dish 2 after 72 hours. Now here both the PQR used, PQR used in both the petri dish and same type of bacteria used, same type of same temperature used, everything is same. Different is here you can see first allow to grow and then place the petri disc. And here straightforward from the beginning, place the disc. Right, now question is, explain why the petri dishes were incubated at 30 Celsius degree. Now what is the answer for this? Why we use specifically 30 Celsius degree? So you might have several options. What is the best thing to get a marks?
So optimal temperature for bacteria to grow.
Okay, one option. Accept. Is the other options? Some written answers. to prevent other microorganisms from entering the medium. That's another option. More choices, more answers. Another answer is lower than body temperature. So bacteria which can affect humans can't grow. Another answer, pathogenic bacteria can't grow. Okay, so we have several answers. Now, uh, when it comes to the temperature, most first thing coming into the mind. Okay, another one, maintain average metabolic rate. Okay, when temperatures come, we always go for enzyme. That's a very common thing. Why this temperature? Because this temperature is the optimum temperature. It affects enzymes. Then you can say enzyme denatured or enzyme rate of reaction decrease. Only for this area, microbiology, you change your answer. Always you can change your answer. This temperature is not temperature of the human body. So then pathogenic bacteria cannot grow. So that's a answer expecting in this place. Whenever you have to propose the temperature, don't propose 37 around, because 37 is pathogenic bacteria can grow, because the bacteria come into our body, they can grow 37. And sometimes I have seen there's an animal and they say this bacteria can grow in human but not in animal. Why is that? And the strong point is because the animal temperature is different. Animal has different temperature, so that it cannot grow. Human 37 is the ideal temperature. Bacteria can grow. Because of that, this microbiology part, this point is very strongly remember to write in microbiology. Human body temperature, if you use 37-degree, pathogenic bacteria can grow. So we should always go below 37 when we are doing experiment. Otherwise, enzyme is the main thing for with the temperatures, but only we change here if you ask this question. Explain why there are more risk involved in the procedure used for petri dish 1 than the procedure used for petri dish 2. Okay, now according to this procedure, this has a more risk high compared to this. Now correctly identify why uh, dish 1 has higher risk than disk 2. These things are quite important, critically thinking such questions. You should prepare your mind now to analyze this kind of a critical questions to give correct answer. Give a little bit of time. So I got a 1 answer, 2 answer.
So petri dish one is opened after incubation, so like it can get contaminated.
Okay, okay, I think yes. One answer is contamination. Another answer because once the petri dish is open to the air, the sample can get contaminated. Yeah. Here you see. Can you remember that certain thing when you buy for our consumption? They clearly say after open this one, keep in the refrigerator. Why they say like that? If we open it, let's say milk packets like this. So once you open it, there's a chance of get contaminated. If we keep outside, the temperature is good. Just after 24 hours, this could be a quickly they grow. If you keep in the refrigerator, the colder temperature cannot increase the number. Okay, here you notice we manipulate two times, petri dish take it out and done it, and again second time we open it. We take it again out and we place the disk in the second time. That means compared to the dish two, the higher chance of get contaminated because we open two times. Okay, just a next one is explain the result of this investigation. Now this is the result. So first one we can see only a one uh, one Q is effective, and for this one P and Q effective. Now think about same bacteria, PQR same antibiotic, but why we see two different results? Think carefully. Why we have this result? So again, I will show you how it did. Take the bacteria, spread on the petri dish, and first one directly incubate 24 hours 30 degree. Second one antibiotic is placed immediately and incubate 72 hours. And here after 24 hours they place the disc and then incubate. I think time is running out, so no one can come up with the solution.
So
Yes,
maybe because the the medium was changed by the antibiotic while they were growing, and since the changes in the medium caused them to not grow properly.
there is no change of the medium in the procedure. Medium means you mean the petri dish? Petri
Yes,
dish.
the agar medium,
No,
like
agar
maybe
medium
the pH
remains
was.
same. It's same, no change. Agar is same. Petri dish is same. Bacteria is same, and PQR is same. Only difference we did, we just 24 hours allow them to grow and then place the disk. Other one immediately we placed. Okay, let's see some written answers. There are more bacteria in petri dish too after contamination and aerobic respiration.
The antibiotic,
Yes, antibiotic.
the antibiotic didn't have enough time to have an effect on P.
Which one? Which antibiotic? Name?
Uh,
Yeah, so maybe like P remain there a longer time in petri dish too.
yeah.
Okay, we come to the solution now. When if we grow bacteria on this one, so 24 hours, if we grow the bacteria, what we can expect, we expect, here I take the highlighter. So then this petri dish completely colonized with the bacteria after 24 hours. Now this petri dish, two, we just spread the bacteria like this, and then we place the disc. That means there is not much bacteria grown when we spread it. After 24 hours, we have enough bacteria. Then look at this difference of these two, P and P. Now I place antibiotic P here. Then again I grow bacteria here. After growth, I can see like this, completely occupied like this, and here this one is, and again clear zone of this.